one, Rajeshwari has messaged that she's in the waiting room. Can you see her? I can't see her. Yes.
Nobody is in the waiting room right now. Actually, we don't have a waiting room.
Dr. Harshita, is it fine now?
Yes, Dr. Dheeraj, it's fine now. Thank you.
Are we ready to start, Surat?
Yeah, just one second. I'm just sending the link to one of the panelists again. I think they're having some issues in joining.
Okay, that's fine.
Just give me 1 minute.
Sure.
So we can start now. Thanks everyone for joining today for our webinar. I am Surat Parvatam, and I'm currently working as the Senior Strategist at Humane Society International India. And I would first like to thank all the speakers, panelists, and participants who have joined us today for this webinar. And specifically, I would like to thank Biotechnology Industry Research Assistance Council, BIRAC, that has been set up by Department of Biotechnology, Government of India, as an interface agency to strengthen and empower emerging biotechnologies in India. Also, STEMPeers, which is one of the world's biggest network of STEM professionals dedicated to create a peer-based mentoring and support platform. And also Center for Predictive Human Model Systems, a policy center that was set up in collaboration between AIC, Atal Incubation Center, CCMB, and Humane Society International India, which is a policy center that has been working towards enabling human relevant research in India. So we would like to start the session, the entire webinar. As you might all know, the topic of the webinar is the need for developing standards for microphysiological systems and the need for having increased funding to develop and accelerate the development of these methodologies, both in India and globally. And we would like to start the session by conducting a small poll. This poll is intended to understand all the participants who have joined to about their awareness about the area of organoids, organ-on-chip, and all these emerging non-animal technologies and the standardization process of these technologies. This is just purely for us to understand what should be, the future verticals for us to concentrate our initiatives on. So the poll questions would appear in your chat box and they would be open for the next 5 minutes. So I would request everyone to check their chat boxes and kind of just fill in the poll questions. So please request everyone to submit their responses and then we'll start the sessions immediately after that. So we'll have the poll open for the next 3 or 4 minutes. So
Oh yeah, I've gotten through my phone.
So building Center for Excellence is one option.
So, we just close the poll in the next minute. So would just request everyone who hasn't filled in to just fill in their responses so that we can start with the session. Okay, so I think we'll start with the session now. So first, before we start the session, I would like to invite Helder Constantino, who is the Director of Research Policy at Humane Society International, to give just a very brief overview of the kind of work we have been doing at HSI to enable the use and development of non-animal methods in biomedical research. Over to you, Helder.
Thank you, Surat. Let me just bring my slides. So I'm hoping everyone is seeing that. Yeah. So hello everyone, and thank you very much for joining us today. Good morning, good afternoon, or good evening, depending on where you are on the planet. My name is Helder Constantino, and I am the Research Policy Director of the Research and Toxicology Department of the Humane Society International. HSI is the largest organization in the world dedicated to promoting animal welfare in more than 50 countries. And today's event is being hosted by the HSI India office. I would like to open this webinar by briefly introducing the Biomed 21 collaboration as the main organizer of this event, and then we'll also present the program for this webinar. The Biomed 21 collaboration is an initiative that was launched by HSI specifically to promote new approach methodologies, or NAMs. We are a global team of scientists and policy specialists who are passionate about scientific progress, and we believe that NAMs can offer researchers new and powerful tools to tackle the major human health challenges of this century, while putting science on a path to eliminating the use of animals in laboratories. We believe that the development of NAMs as mainstream tools in biomedical research will require a system thinking approach. Indeed, the development of new and emerging research models intersect with very different scientific and policy areas. The fast pace with which new technologies based on human biology are reshaping basic and applied biomedical research calls for proactive policies able to support this evolution in fields as diverse as drug discovery, personalized medicine, research funding, biobanking, regulation, or training and education. And that means that finding the way forward will require the support of various institutions and stakeholders working collaboratively to craft data-driven policies. Therefore, dialogue and inclusion are core values of our work. And in that regards, I would like to especially thank our engagement partners today for this webinar, the Biotechnology Industry Research Assistance Council, BIRAC, of the Government of India, STEMPeers, and the Centre for Predictive Human Model Systems. I would also like to welcome the speakers, Dr. Ewart from Emulate, Dr. Majumder from the Indian Institute of Technology. That's for the first session. Session 2, the panelists will be Dr. Becken from the Belgian Federal Agency for Medicines and Health Products, Dr. Stacy from the International Stem Cell Banking Initiative, Dr. Adi Seshan from the Bill and Melinda Gates Foundation, Dr. Gandhi from BioRx Venture Advisors, Dr. Kumar from BIRAC, and the session will be chaired by Dr. Gupta, who is also Vice Chair of the National Standing Committee on Medicine and Healthcare Products. What brings us today specifically is our microphysiological systems, also referred to as MPS, such as organs and chips and organoids, which have experienced a tremendous development in the last decade and show great promise in bringing human biology to the core of biomedical research. However, there are also many challenges to their development, and this webinar aims to move us forward to overcome these challenges, and we will approach this discussion in two parts. Session 1 will cover the standardization of organ chips and will be moderated by Dr. Pistolato, while session 2 will be a panel discussion on funding policies to accelerate the development of MPS, and that session will be chaired by Dr. Gupta. Thank you very much again for participating to these events. It's great to have you here, and we look forward for a very stimulating and enriching discussion today. Thank you.
Hello for providing this overview. So, and now I'll just briefly talk, I'll just show like a few slides on why exactly we are conducting this webinar and what is the topic that we are interested in today. So I'll just share my slides. Today, the topic of our discussion is going to be why do we need globally harmonized guidelines for microphysiological systems? But before we kind of move on to that, for people who might be joining this webinar with who might have heard about this entire concept of human relevant models for the first time, I just want to briefly talk about what exactly are we talking about and why is the need to talk about this subject. So human relevant models are systems, they could be either in vitro, computational systems that are based more on human biology and human cells, in contrast to animal biology or animal models. So these would include in vitro systems such as organoids, organ-on-chip, or they could be advanced imaging techniques which allow you to directly image human biology, or it could be computational too. And for today's webinar, like when we say microphysiological systems, we mean either both organoids and organ-on-chip systems, which is what the systems that we are going to be talking about. And these systems, the human relevant models have given us a tremendous insights in the last decade or so. For example, organoids were very useful to understand how exactly the Zika virus modeled human brain development and caused microcephaly. Similarly, organ-on-chip devices provided great insights for developing COVID-19 therapeutics. And in cognition of the great potential that these systems have, several global regulations have been happening across the globe, both in India and different countries. For example, in 2019, US EPA announced its plan to phase out the use of mammals to test chemical safety by 2035. I would like to point out that January 2024 this year, this hard deadline of 2035 has now been removed. EPA has still commented that it's committed in its plan to phase out the use of mammals, but they have removed this hard deadline. Similarly, in 2020, the promotion of development of alternatives to animal testing, or the PAM Act, was introduced in South Korea. In 2021, the European EU Parliament passed out a resolution to phase out animal testing through a dedicated action plan. In 2022, US passed the landmark FDA Modernization Act 2.0, which allows the use of non-animal methods for drug safety and efficacy testing. And then the WAM Act was introduced to supplement the PAM Act already in South Korea. In 2023, several advancements happened, like Brazil banned cosmetic testing. India also passed its landmark amendment to new drugs and clinical trial rules, wherein even in India, now one can submit non-animal methods data for testing drug safety and efficacy. Canada passed the bill S5 to reduce reliance on animal testing and chemicals. And similarly, UK government also clarified that animal testing was not mandatory for testing new medical products. And European Commission also introduced the new EU pharmaceutical legislation was proposed, which again encourages the use of non-animal methods for testing of medicines. And this start one second. In 2024, the FDA Modernization Act 3.0 has been introduced, which again facilitates the use of non-animal models for drug safety and efficacy testing. And okay, and the use of microphysiological systems has also seen a great increase in adoption by pharma companies. In this table, you can see all these different industries who have already adopted microphysiological systems in their internal portfolio decision making. However, despite these advanced adoption by industry and also these global regulations, the uptake still has been slow. And one of the reasons that has been recognized is the lack of globally harmonized standards. By standards, I mean that different technology developers, whether it be pharma or academic, they have their own set of standard operating procedures or protocols via which they develop these organoids, organ-on-chip devices, which makes it difficult for increasing its adoption across different stakeholders. And standardization has been realized as a major enabler for innovation. And if we look at specifically in the context of organ-on-chip, for people who might not know what exactly is an organ-on-chip device. So organ-on-chip device is a AA battery-sized chip, which you can see on the left side, and it contains these microfluidic channels, which are lined by epithelial cells and endothelial cells and sandwiched, and there is a semi-permeable membrane sandwiched between the two layers. And it also has a flow, which is meant to mimic the fluid flow inside our body. And this kind of organ-on-chip system has both technical or the engineering aspects, such as the fluid flow, how the fluid flows inside our body. It also aims to mimic that. Then there are environmental conditions. There are sensors in this chip which detect various oxygen, pH levels, etc. And then there are biological aspects, such as what cells you use, what biomaterial you use, what kind of phenotypic characteristics of the biological model are you able to replicate in your system. So an organ-on-chip device would have two aspects, and it is understood that standardizing biology, it may be difficult because of the nature of the biology itself, but what you can standardize is how the various procedures that you use it to model this entire system. That is something that we can begin to work towards. And the various standardization needs include, the first and foremost is defining the context of use. So each technology developer that develops an organ on chip, for example, a liver on chip. So there isn't a universal liver on chip that you can use to address all liver related questions. So each organ on chip is designed with a specific question in mind. So that context is very important to define before you begin to develop your system. Another, there is a need for developing standard protocols and criteria for the technical functionality, the biological functionality. How are you going to assess the biological relevance, robustness? So it's very important to define all of these aspects. And these standards need to be robust, reliable, affordable, and also incorporate all the perspectives of end users and stakeholders. So we recently collaborated with stakeholders across the globe, and we published this policy paper, which exactly talks about this aspect, that these standards and protocols and guidelines need to be harmonized globally, so that each country does not have its own set of guidelines, or each end user does not have their own set of guidelines, because having these globally agreed upon set of policies and standards would help to ensure wider adoption of these systems across different stakeholders and also increase confidence in these systems. So that's what we are going to be talking about today. So with that, I'll so with that, I would like to call upon Dr. Francesca Pistolato, who will be moderating our next session. So Francesca joined HSI last July as Senior Strategist for Biomedical Science. She previously worked at the Joint Research Centre of European Commission, where she contributed to the development, optimization, and dissemination of human iPS-derived models for developmental neurotoxicity testing. So before I hand over to Francesca, I would also like to ask all the participants to type in whatever questions they might have during the session in the Q&A. There is a Q&A box. So, and we can take those questions at the end of each session. So with that, over to you, Francesca.
Thank you very much, Surat. Good morning, good evening, good afternoon. So in this session, we have two very well-known speakers who will talk about the standardization aspects of their organ-on-chip devices. Let me first introduce Dr. Lorna Ewart. She is the Chief Scientific Officer at Emulate, a leading biotech company that has commercialized organ-on-chip technologies. Dr. Ewart focuses on leading scientific strategy, guiding product development and commercialization of next-generation in vitro biological models, ensuring an execution of bespoke contract research studies for biotechnologies and pharmaceutical companies, while also establishing collaborative external relationships with academics and regulatory authorities. She is passionate about changing the drug discovery process through adoption of human-centric translational models. So now the floor is yours, Lorna. Please.
Thank you very much, Francesca, for the kind introduction, and also to HSI for hosting this event and inviting me to participate. I have a few slides to just set the scene. Also, I know that the next speaker will do likewise, and I very much welcome the Q&A that will follow after the short presentation. So with that, I shall just... Oh, goodness. It's not going to allow me to share my... Hang on a minute. I should have tested this beforehand. My mistake. This happens, doesn't it, with updates to your computer. Just 2 seconds. It will ask me to... I need to just drop off and rejoin. Just give me 1 minute. I'm sorry about that.
It's okay, Lorna.
There we go. Okay, perfect. Francesca, can you confirm that you can
Yes.
see my desktop, please?
We can see it.
Wonderful. Okay. So really what I wanted to do in the next 5 to 10 minutes is just share a few perspectives from Emulate in our journey towards organ chip development. For people who are not familiar with Emulate, Emulate was a spin-out organisation from the Wyss Institute at Harvard Medical School in the United States of America. It became an organisation in January of 2014 and started commercial activities in 2018. And fundamentally, our goal is to design and develop both the microfluidic platform as well as the biological applications on the organ chips themselves, with a view to accelerating the drug discovery process in a more human-centric and relevant manner to avoid and reduce the use of animals in this process. So the way that we think about this at Emulate is through these 4 guiding principles, and we call it designing for the unknown. So when you speak to our engineers, they have a very nice turn of phrase, and what they say is that the instrumentation, the actual engineering side, is the stage for the biology to shine. And as a biologist, I really appreciate that analogy. But of course, to get to the instrumentation in the first instance, as well as the consumables, which you can see here on the left-hand side of the diagram, that there is a very close interaction between engineering and biology. And having come from the pharma sector myself, I liken it to the conversations that need to take place between a chemist and a biologist to develop a drug. Likewise, you need good conversation and understanding between engineers and biologists to generate this unique platform as well as the biology, and thus allowing it to shine. So the way that we think about this at Emulate is that our instrumentation should be what we call a wide solution vector. In other words, we want to have a single consumable, which you can see on the left-hand side, at least a single consumable footprint, which will ultimately enable us to place different biology within it. And therefore, the instrumentation has to be adaptable to enable that different biology. So, for example, our platform will apply a microfluidic flow to the cells, which, as Surat mentioned in her introduction, is an important stimulus to the cells in terms of a shear force. But uniquely to the Emulate platform, we also apply a vacuum stretch, which is important for modelling organs such as the lung, particularly down at the alveolus, the intestine to mimic the peristaltic motions, or indeed the skin, which is actually in most places of the body under continual stretch. We also have, when we're designing our instrumentation and our consumables, we have to think about how we can commercialize our products as well. And that really leads into some of the discussions that we'll have today around standardization of our approaches, our biology, our methods, but also certification around standards of the instrumentation itself and quality assurance on the consumables. And lastly, and perhaps most importantly, this idea of only add complexity if it's needed and only when it's needed. So our founding scientific director, Professor Ingber, will always talk about try to do something as simply as you can. And if you can't recreate the biology you're looking for, start to add complexity. So for example, in some of our more recent models, in fact, we launched a model around CAR-T efficacy on Monday there. Our complexity add was to add circulating immune cells to our chips and encourage a physiological recruitment of those immune cells from the vasculature into the epithelial tissues. So our guiding principles from an engineering perspective are just as important as those from biology. And we talk about the abilities, and you can see the 6 abilities here. And I'm not going to go through each of these in turn, but I think there's some key phrases here. So for example, adaptability, we need to be able to adjust our models, our platform, as well as our consumables to meet the context of use that we are developing a model for. And that I'm sure will come up several times in our discussions as well. We also want this idea of extensibility down in the bottom right-hand corner. And this allows us to future-proof our instrument such that users who engage with our instrument through rentals or purchase don't find themselves in a period of time no longer being able to use the platform. Indeed, we're committed to delivering new biology and new models together with platform enhancements. So for those who are not familiar, this is a picture of our fluidic instrument. We call this instrument Zoe. Zoe is the Greek word for life. Zoe can actually support 12 organ chips. She has 6 in one drawer that you can see in the blue handles on the left-hand side and 6 in the other drawer. And she sits within the incubator where she is able to equilibrate with the humidity and the partial oxygen pressures within the incubator at the temperature that it's set as well. Now, the important part here is that our chips will sit in an assembly on the right-hand side that we call the pod. You can see that in the middle photograph there. This is our fluidic reservoir. We have a reservoir for the upper channel of our chip and the lower channel, which I'll show you in just a minute. And we have what's known as a unidirectional flow. So we will flow in through the inlet and then collect the effluent in the outlet. And this allows us to do many measurements over a period of time, adding value to the data that scientists are developing. And here are some more images, just how a user would be setting up the platform for those who are unfamiliar. Number 1 shows the seeding of cells within the chip. You can see the 2 parallel microfluidic channels, with one being injected with a coloured dye to help one visualise it more easily. Next, that chip is connected to its pod, which you can see in picture 2, and that contains the media that will run through the course of the experiment. 3 is the addition of those chips into this drawer. Remember, I said that each drawer contains 6 chips. And finally, you can see the scientist placing that drawer into the instrument, which sits within inside the incubator. So I focus a lot there on engineering. Actually, as people probably know, my forte is actually more in the biological space. And so I get much more excited in thinking about how we create the cellular microenvironment for the particular cells of choice to flourish. And I think for this, it's important to remember that the cellular microenvironment is extremely complex. And recall, we have a philosophy at Emulate that says we want to start as simply as possible. So we start with the addition of flow. We look to optimize our flow rate. Some cells prefer a slightly higher flow rate than others. So for example, endothelial cells usually like a higher degree of shear stress, perhaps compared to a hepatocyte, which is quite fussy. Hepatocytes don't really like a lot of shear stress, and so we find we have to adapt and optimize flow in those cases. I mentioned earlier about the application of a mechanical force, in particular organs such as the lung, the intestine and the skin. Mechanical forces will be important, whereas organs, again, like the liver, it's a solid flat organ. We typically would not apply a strain or a stretch in that situation. Extracellular matrix is incredibly important. We usually apply this to the channels of our chip prior to seeding any particular cell. And what we're looking for is to have a matrix that is familiar to the cell, so usually containing things like collagen, fibronectin, laminins, etc. And that encourages the cells to grow in a monolayer formation and make those important cell-cell interactions that, again, enable the cell to start to believe that it's in an environment that's familiar to itself. In some cases, cell shape and then cytoarchitecture are particularly important. We see this in the formation of 3D tissues in our intestine models, and that's to create that crypt structure that's very important to see those different cell types in the crypt, particularly the dividing cells at the bottom of the crypt, really find a home that they can survive and flourish on the chip. Our chip being 2 channels is simply around creating an endothelial epithelial interface. This is the most basic functional unit of any tissue. So we refer to this as a tissue-tissue interaction. And indeed, us and others have published many scientific papers that show how endothelial cells communicating with epithelial cells drive a more physiological function. When we start to think about complexity, we are now actively looking at involving both resident and circulating immune cells. So again, as scientists, we know that the basic premise of many, many diseases is usually an aberrant immune response. And increasingly with biological therapies being sought after for treatment and in some cases cure of particular diseases, it's very important for us to be able to recreate that circulating immune cell response and allow researchers to assess efficacy of their proposed therapies. And possibly also toxicity as well. We do see, for example, elevation of cytotoxic CD8 positive T cells and liver injury as a consequence of some biological therapies. And lastly, and importantly, our chips are made of PDMS. This makes them oxygen permeable. I mentioned that our instrument and therefore the chip sit inside the incubator. So they'll usually be around 95% oxygen in that incubator. But for some tissues, again, like the intestine, some areas of the intestine do need to create a hypoxic environment. And this is particularly important for some of the microbes that are known to be within the microbiome. So again, we have methods whereby we can control the oxygen gradients within our chip to encourage the greater complexity of adding microbes. Lastly, I just want to mention a few words about the work we've been doing on human liver chip. Here on the left-hand side, you can see a diagram of how we actually construct our liver chip. So hopefully by now you'll understand that our chip has 2 parallel microfluidic channels. They are both fluidically independent. Therefore, we can use media that's optimized for hepatocytes, in this case, that are situated in the top channel, and non-parenchymal cells, which are situated in the bottom channel. There is a membrane that separates these 2 channels. It's also PDMS. That's because we need it to be stretchy for our chips that are stretched, but it has pores within that membrane, which are 7 microns in diameter. This therefore allows good cell-cell communication, but it also allows the passage of circulating immune cells. In the case of the liver chip, we're seeding the liver and the hepatocytes in between two layers of extracellular matrix. This is a very common way of culturing hepatocytes, and it's needed to enable them to correctly polarize so that the important drug transporters, for example, are expressed on either the apical or basal membrane of the cell. In the channel below, we see non-parenchymal cells, which are the liver sinusoidal endothelial cells, a very specialised endothelial cell for this organ, characterised by what's known as fenestrations, or if one thinks about it, it's a very large sieve. So this is why many things will go into the liver, particularly, this is one of the reasons why organisations are interested in using adeno-associated viruses, for example, to treat liver disease. But however, they can also cause toxicity, which is perhaps less desirable if your treatment is in another organ. We also have the stellate cells and the Kupffer cells. The Kupffer cells are the resident macrophage cell line and cells, I beg your pardon, and they're particularly important as a first line of defence. Now, to the topic of standardisation, we undertook a very large study, and we think it's one of the largest studies of its kind. It was published in the Nature's portfolio Communications Medicine in December 2022. And you can see that as of about a week ago, it had been accessed more than 38,000 times with 60 citations and an altmetric score of 750. So this in itself is evidence of not only the interest in organ chips or non-animal methodologies and the ascendance of such for the pharmaceutical industry, it's also indicative of how one might undertake such a large study on a first step towards a qualification of this particular model. So what we did here was we used just over 870 liver chips. It was a mammoth undertaking. We used the chips over 5 experimental cycles. We used 3 human hepatocyte donors, and we tested 27 small molecule drugs, which were known to either be toxic, e.g. showed drug-induced liver injury in the clinic, or negative. Now, importantly, we set ourselves this experimental design using guidelines produced by the IQ-MPS Consortia. This is a group primarily based in the United States, although it does have representation from European pharma companies. And they set out some guidelines and some steps for organisations such as Emulate to build a model. And importantly, they also put forward 20 of the small molecules that we tested. So in other words, they were asking particular questions of the model with these small molecules. And what we found in this particular study is that our chips were able to detect toxic drugs with an 87% sensitivity. So for every 7 out of every 8 drugs that we tested, we correctly called them. And it also had a 100% specificity, which means that for pharmaceutical scientists, the likelihood of discarding a drug because it was flagged as a false positive is 0%. And lastly, and importantly, we showed in comparison to animal models, and this animal models did allow these drugs to go into the clinic, as is still the case today. Animal model experimentation is required in safety to move a drug into first-in-human trial. However, if the animal perhaps didn't, in some cases, get this risk assessment correct, because of the drugs that we tested, we know that several of them were associated with patient fatalities or indeed liver transplantation. So we would advocate that it is important to think about how one begins to characterise the model. And I'm very much looking forward actually today to talking about some of the standards that we've been adopting in our biology as well as in our engineering, and how, of course, those could help the field progress. And with that, I will stop sharing and pass to the next speaker.
Thank you very much, Lorna. Amazing, your work, amazing what you're doing with Emulate. Actually, we think it could be a good idea to take a couple of questions addressed to you, if you don't mind. So one participant was wondering about wait a second, where is it? Here. So about the significance of organ on a chip in association with precision medicine, can you comment on this?
Yeah, absolutely. So many organ chip developers do talk about precision medicine, and really for me, that is definitely a goal. We talk about it less at Emulate simply because we're focused on building robust, reliable and reproducible models. But simply the goal would be to use perhaps patient material in the models themselves and start to really think about how to target particular patient groups. So for example, in certain cancers, you would maybe want to be characterizing that particular cancer on the chip and understand which drug, either alone or in combination, could offer particular benefit to that patient population. So I think it is important going forward that we think about how to access more patient samples once we're convinced that we have good, solid, robust, reliable and reproducible models at baseline. So I think we will start to see more of that as we progress through, and hopefully that also contributes to the reduction, of course, not just in animal usage, but also in clinical trial attrition, because companies will be able to target those disease cases with the most precise or accurate regimen that they possibly can.
Thanks a lot, Lorna. I would actually take just another question, then we can allow Dr. Abhijit to give his presentation and take additional questions after his presentation. So for you, Lorna, have you guys looked into hydrophobic drugs and the issues they have with PDMS?
Yeah, we have indeed. Thank you for asking that question. So I did mention that our chip is made of a rubber polymer, PDMS, and for people who are perhaps less familiar, this polymer is known to cause drug absorption, particularly of small molecules that are lipophilic in nature, usually with a known C log P around 2 to 4 in molecular weights that are, of course, common with drugs that are being designed for oral bioavailability, as well as certain characteristics around polar surface area, for example. So it's actually quite a complex topic, but quite simply, yes, we are aware that PDMS could potentially be a limitation. What I would tell you is that we did not find that in our assessment of 27 small molecule drugs. In part, that is because we are able to have some workarounds. So, for example, in some cases, we did increase the flow rate a little bit to ensure there was sufficient drug delivered to the cells over the course of our experiment to see the toxicology. Now, we are aware that for some particular applications, particularly those involved in drug transport, which are usually looking acutely between the 2 to the 4 hour time point, this might not be a particularly good workaround, and therefore we are working on similar consumables that do not have the PDMS. Of course, the flip side of that is that PDMS is stretchable. So we need to think of ways in which we don't reduce the functionality of the chip, and also other plastics, of course, have much more limited abilities for imaging as well. So it's a very complex topic, and I'd be very happy to follow up the individual and talk more. It's certainly an area that we're looking at extensively right now.
Thank you so much, Lorna. Very insightful. I would now actually start by introducing Dr. Abhijit. So Dr. Abhijit Majumder is a professor at Indian Institute of Technology in Mumbai. He did his postdoctoral research in cell mechanics and microfluidic devices. He likes to call his lab as the M lab, which captures the 4 major areas of work: materials, metrics, mechanics, and microfluidics. The lab is trying to mimic cells in vivo environment as much as possible using systems such as organ on a chip. Dr. Abhijit Majumder also has keen interest in spreading scientific awareness among masses, particularly students. And with this, I would like to give the floor to Dr. Abhijit. Thank you.
Thank you, Francisca. And let me share my screen. Am I audible?
Yes, You are perfectly audible.
And is my screen visible?
Yeah, you're sharing your screen.
Yeah.
Yeah. Now also the
And
presentation.
it is fantastic. So yeah, I guess my previous speaker has made my job a lot easier, because she has fantastic, she has explained the need and the present commercial and the industrial standard. So what I will do in next 15 minutes, I will try to talk about something that our lab is working on a particular, the organ on chip, and that is called the placenta on chip. And this is, we are working in collaboration with Dr. Deepak Modi from NIRH ICMR and Dr. Debjani Paul from PSB IIT Bombay. So when we talk about the placenta, this is probably, arguably the most important organ, because our life starts from there. So just for the, to bring everyone on the same page, placenta is essentially a gatekeeper that helps to protect the developing fetus. It allows to transport oxygen, carbon dioxide, nutrients, but at the same time it protects the developing fetus from various toxic element, immune cells, and other microbes, bacteria, viruses, etc. So this is a very important organ, and any kind of problem in this placental development will cause maybe the miscarriage or the defect in the development, etc. So when we look at the placenta, this is essentially the umbilical cord that is going to the fetus. From there, we have the fetal vein and the fetal artery, which is getting divided into these tiny villi-like structures to increase the surface area. And then that gets open to its large pool of blood, maternal blood, which is here. And this large pool of maternal blood is coming through the maternal artery and the maternal vein. So if we look more closely here, which is essentially working as the barrier, we have these capillaries, which is the fetal capillaries, and then we have the trophoblast cells, which creates this lining. So one side we have the endothelial cells. In the fetal side we have fetal endothelial cells. Then we have the fetal capillary, where we have the fetal blood. The other side we have the trophoblast, and then we have this pool of the maternal blood, which is the intervillous space that we can see here. So any drug, any chemicals, or oxygen, carbon dioxide, etc., that gets transported or transferred between this fetal blood and the maternal blood, passing through this barrier made of predominantly these syncytiotrophoblastic cells, fetal endothelial cells, and some ECM layer. So this is something that we would like to develop or to mimic to check the transport properties. But before going there, let me also kind of discuss that compared to other organ on chip models, why the placenta on chip model is so critical and why we are so much interested in developing this particular organ on chip. When it comes to the medicine or approval of the medicines clinical trials, the pregnant women, they are excluded from the clinical trial because of obvious ethical reason. But at the same time, that creates the problem that out of all the available drugs that we have, which are safe for the a pregnant woman and which are absolutely unsafe, this understanding is very poor. We have only a list of few drugs that we know at this moment that safe for the use for the placental for the pregnant woman. And this is very important because there can be the pre-existing conditions such as TB, epilepsy, For that, the mother may need to take the drug, and the problem is that we don't know whether the drug is safe or not. And as a result, many of the times the clinician, they stop the using of that drug, and which may have the other kind of effect and other very serious effect during the pregnancy. There are various different, the conditions that develop during pregnancy, such as preeclampsia, gestational diabetes, other various different discomfort. For that, the mother may need to take the drug. And again, we come back to the same issue that we don't know which drugs are safe and which are not. On top of that, when we look at, there are this study that is happening at Boston since many decades, and it has been shown that 45% of the pregnant women, they actually take the over-the-counter drugs, and for which many of them, they are not classified as safe for the pregnant woman. So as a result, we need to have a model to know that which drugs, at least to first step to know that which drugs or which chemicals may cross the placental barrier or not. And again, coming back to the problem of ethics, that we cannot do the test on the pregnant woman. The other option is the animal testing. Again, as we all know in this, this particular audience, that the animal testing has its own various different issues, particularly in terms of its predictive ability. That those issues become even more critical when we come to the placenta, because the placental structures of different species are very different. For example, when I talk about the human placenta, as I already described, there are essentially two cell layers, syncytiotrophoblasts and the fetal endothelial cells. But if I take a rodent model, there are 6 different layers. In 6 different cellular layers. If I look at the placental structures, here the fetal capillary is actually kind of floating in a huge pool of maternal blood. Structures are also not there in many of the animal models. The closest model is obviously the primates, but then again, doing all the tests in the primates, they have its own issues. So here comes the importance of developing the placenta-on-chip models, and we are not the first one to develop it. This is 2023 numbers. That number has changed now. So there are like now kind of 17 such published paper which has talked about the placental barrier. So the basic structure in developing the placental barrier is that you have a membrane. In one side of the membrane, you have endothelial cells coming from fetal endothelial cells. Other side, you have maternal trophoblast cells, and you try to mimic the transport. And these are the various different things that people have checked, like the glucose transport, caffeine transport, effect of bacteria, various different drug transport. And one of the disease model, that is the preeclampsia, which is again very common in the pregnant woman. That model has also been developed. What is kind of missing is this in vivo like transport. I will talk about that when I will talk about the importance of standardization. So in our lab, what we are trying to develop is kind of two different models. One is that high throughput models, that is, it is not with the flow. We understand that the flow is important to mimic the transport processes, but here we are making it flowless so that it can be high throughput for the initial screening of many drug or the potential drug molecules. And also we understand that the perfusion will increase the transport processes. So whatever drugs that cannot cross the placental barrier in our placenta-on-chip process in the flowless, that probably we don't need to take for the perfusion one, but which crosses that we may now take in the perfusion, and we can take look further. So I will not go into the details of the structure of our devices, etc., because that is not the purpose of this talk. However, we will just show that how we are standardizing or trying to standardize our device. So one side we have the vivo cells, which is the trophoblast cells, and other side we have the HUVEC cells. In between we have a membrane, and this is here you can see the cross-sectional view of both the cells. Here you can see that they can make a tight monolayer that is very much important to create the placental barrier. Then this is the trophoblast. Vivo is the trophoblast that we put, but then in the placenta what we have is the syncytotrophoblast. So the syncytotrophoblast is that these trophoblasts, they aggregate and fuse to make a large kind of a cover of the cells. So that happens in presence of a chemical called forskolin. So here we have treated our vivo cells with forskolin, and we have shown that they are forming these fusion and for creating this syncytized trophoblast layer. Also, when this layer forms, they are known to secrete ACG that we have checked using the simple pregnancy test kit. So that kind of tells us that, okay, structurally our system is mimicking the placenta. Then to look for the transport processes, we have checked here 3 different molecules, and we are now checking other drugs. First, we are talking about FITC, which is a fluorescent molecules, a very small, small molecular weight. So they are supposed to cross the placental barrier by simple diffusion. So here you can see that we are comparing the membrane without cells and the membrane with the co-culture of endothelial and syncytized trophoblast. And you can see that for the small molecules which are supposed to pass the placental barrier, our co-culture is not causing any extra hindrance or resistance to the transport. But when we use the high molecular weight dextran, so FITC dextran, these are the large molecules, so they are not supposed to pass. And that is what we see here, that in the fetal chamber, when there are no cells, just membrane, this FITC dextran can pass, and so in the fetal chamber the concentration of the FITC dextran increases with time. However, when we have the placental barrier, this FITC dextran, being the large molecule, it cannot pass. On the other hand, in case of the glucose, there we have the facilitated transport using the GLUT receptor. So there it gets transported from the maternal chamber to the fetal chamber. However, to a certain extent, not as the rate of transport is not as large as just without the cells. So here the cells are helping the facilitated transport, but with some control. We have also tried to mimic our device using some simulation, and we show that our simulation result and in our experimental result, they match. So now I will come to the last slide of my talk, and where we are talking about the challenges in development and particularly the validation. So here what we are looking at the transport properties, and as Dr. Lorna said just before me, that the shear stress plays a very important role, and particularly in this case, even in the placental biology, it has been shown that the shear does play a very important role how the cells mature. So, but the question comes that when we mimic the placenta into a micron size device, how do I scale so my transport properties remain the same? And if we look at the literature, as I said, there are 17 kind of published paper. The flow rate varies from 0.6 to 1300, so wide range. So one is more physiologically relevant? Again, the question comes that how to standardize and which one should use when they, design their own placenta on chip. As Surat said, that it is always context specific. Still, we need some kind of standard here. Also, the placental structure changes during the pregnancy. The layer becomes thinner. The expression of influx and efflux pumps changes. As a result, the active transport process gets modified as the pregnancy goes from first trimester to the second to third. So question comes that which stage of pregnancy we are mimicking. Relevant cell types, because most of the work that has been done and also our work, we are using the cell types that are taken from the choriocarcinoma patient, which might not be the exact representation of a healthy placenta. Also, we need to think of the ratio, at which ratio we need to use the cells. Lorna mentioned about the ECM, the chemistry and the mechanics which type of ECM, all those play a very important role. Then when we talk about, if you remember, we talked about that we are putting these two cells in two sides of a membrane. Membrane pore size, membrane material, that also plays an important role. And again, when I look at the literature, the pore size people have used varies almost an order of magnitude. And then as we are talking about the context, so each design might be very context specific, that which stage of pregnancy I am mimicking. I am mimicking a healthy placenta or a diseased placenta. Am I mimicking a placentopathy? So what I am mimicking, that also is very important. And finally, we also need to think of the population difference from a Caucasian population versus other populations. Are there any difference? And then what we design for, what we standardize it against which population, which which genetic makeup. So, and finally, when we talk about the drug safety does not depend only on transport. It also depends it does not only depend on yes or no. It also depends at what concentration it is getting transported. So finally, if we need to really validate it, we need to validate it against human data. But if I go back to the square one, that we don't have much human data because of the lots of ethical issues in this particular context. So that's why I think this whole meeting is very important, that for each of these different organ-on-chip models, the global community needs to come together and to discuss that what should be the standard, how one can say that, okay, this particular organ-on-chip model can be used for this particular context. So I would like to thank my lab members, the people who actually did actual bench work, Saurav, Anshul, Sheetal, and Yash, my collaborators, Dr. Deepak Modi and Dr. Debjani Pal. This whole work was funded by Imprint, DST SARB Imprint. And when we talk about the funding agency, obviously it is funded by the citizens of India thank you to all of them. And thank you for your attention.
Thank you very much, Dr. Abhijit. I would now quickly go to a few questions that we have received here in the chat. There is a first one from Dana Connors. This is actually addressed to both of you guys, so Lorna and Abhijit. So maybe we can give the floor first to Lorna and then ask Abhijit to provide the further inputs. So can you speak to the replication of system impacts at the cellular level with MPS? How do we make these whole organism representative and how do we standardize them? That's an interesting question when thinking about standardization. Lorna?
That very is an interesting question. So we've focused a lot on our liver model, and one of our major readouts on the liver model is albumin production, for example. So what my team do is they are watching those albumin levels keenly across chips within the study, as well as chips across studies. And using that, we can start to build up a picture from that particular donor. So maybe I just qualify that to say we have different hepatocyte donors. So different donors, as you can imagine, will produce albumin at slightly different amounts. So therefore, we never say X amount is the amount, if you will, and we begin to characterize our donor and understand how it performs. And then the team are looking for the chips that they have in each study, how at baseline they're performing. Are they within that range we would expect for that donor? If not, why not? Occasionally we've seen them be a little bit higher. Sometimes you see them being a little bit lower. Usually when it's a little bit lower, it's a technical problem, maybe a small bubble, for example. We are still not sure about the higher, though. That's only been seen once or twice, but any ideas there would be very welcome. But then we plot that over time as well. It's a very standard statistical way of looking at your outputs. It's often referred to as a Manhattan plot. So when you plot your vehicle values, pick a day or pick several days across the study, and then you're monitoring. And if your vehicles are changing from study to study, that often tells you something's happening to the model. It could be something to do with the stability of the cell. It could be something in the way that the model itself is behaving, and you're looking for as flat and as straight a horizontal line as you can possibly get, and we internally are seeing around about 96% reproducibility. So in other words, we're getting that horizontal line about 96% of the time within a particular donor. So that's how we're looking at it, and we're trying to think about how we can provide further guidance to users to say, if you're out with that particular range, do X, Y, and Z. So that's some things that our team are thinking about to ensure the customer is successful in their studies. But in terms of standardization, I know from a biological perspective, endpoints, standardization is important. So albumin production from a hepatocyte is a very good one. Most people will align on that being a good marker of baseline functionality of that cell. So that could be a good place to start in terms of saying for any hepatocyte to go into these models, it needs to be between X and Y range across, A to B time frame, and showing that at least 95% of the time or greater. So you can start to see how these standards may evolve.
Thanks a lot, Lorna. Abhijit, any insights with regards to standardization in the context of placenta-on-a-chip systems?
So I guess, Lorna has covered the basic principle, and that is what we also try to show in our placenta-on-chip devices, that there are the certain functionalities that at least we need to show that, okay, these are the certain functionalities. For example, as I showed that the HCG production or the fusion of the cells. And as far as the transport processes are concerned, there are, I mean, again, I am giving the answer from the placenta-on-chip point of view, that at least for some of the drugs, we know that, okay, they cross the placental barrier. Caffeine, we know that it crosses the placental barrier. Thalidomide, we know that it crosses the placental barrier and cause harm to the fetus. So those kind of already known drugs, and the drugs which are also known to be safe, very few, but so those can be used to show that, okay, this particular device is it can mimic to a great extent what has been observed so far.
Thank you very much. Very interesting.
Francesca, can I just build off of that point?
Sure.
I think that's important as well. I focus my answer on the cell baseline, but clearly if the context of use is known and there's reference drugs that are known to either cause a response or not, again, that's another thing that can be standardised. And I know that that's widely discussed as being a critical area of alignment in the community for certain contexts of use to get that test set spot on, and that will also enable more of a, when you've got different donors, for example, you're always looking, you still want to understand whether they're going to show the effect or not. So yeah, I think that's a really important point to bear in mind.
Very good point. Thanks a lot, Lorna. For the sake of time, I think we can take probably this additional question. I would actually invite you, Lorna and Abhijit, to reply to the remaining questions if you think you can provide any answer directly in the chat or in the Q&A function. So, but let me ask you this thing to both of you, if you can provide a sort of quick reply, hopefully. I think it's an interesting one. So OECD guidelines have test guidelines for human reconstructed skin, this, for example, skin sensitization, etc. So this test may help validate the skin models, these more complex tests. What about other organs? Why are we facing challenges in establishing test guidelines and regulations for in vitro models?
Abhijit, do you want to go first?
Actually, not kind of, there were some connectivity issues, so I'm trying to read. If you can go first, I will just join.
Absolutely, no problem at all. So whoever asked that question, that's my question as well. So, but I would probably say, in answer to that, one thing that I've learned over the last 18 months to 2 years is that something like an OECD guideline doesn't occur overnight. So it's very much a long process, lots of discussion, lots of involvement from regulatory groups across the globe, because as you mentioned, it is a global guideline. So I would say that maybe the reason we have not yet seen it is more a timing phenomena rather than a reason that the OECD, for example, do not want to give a guideline for such model. So I am very optimistic that we'll see those discussions initiate across the coming years, and maybe we can circle back on this conversation down the line, and hopefully we'll talk about the first guideline for a model that has been there. So but that would be my personal view, and welcome any other insight.
Thanks a lot. That would be fantastic.
So may I just add
one
point here?
Sorry. Yeah, go ahead.
So I think the skin model is the easiest one, because the medicine or the other chemicals that we are testing, that is not going to the system. So it is kind of an external application. So the quote unquote, the risk is comparatively less compared to say a liver-on-chip model or the placenta-on-chip model, because if our model system fails to predict, that will have a much more serious consequences. So I guess that is also, I mean, what Lorna said, I fully agree with her. But on top of that, I think this is also another feature that why the other organs are kind of taking longer.
More, higher biological complexity, different cell models and cell types included. Okay, I see. I don't know, Surat, shall we take an additional question, or for the sake of time, shall we move on to panel number 2, to the second phase of the webinar?
Yeah, in the interest of time, we can move on, but I would still, ask the participants to type in whatever questions they have in the chat box. And if I would request both Abhijit and Lorna to answer the questions in the chat box in the interest of time. I mean, there are a few more questions, but because of the interest of time, we'll have to move on to the next session.
Yeah, happy to do that. Thank you again for a nice session.
Thanks to you, Lorna and Abhijit. Very, very interesting.
Thanks, Francesca, for chairing the session 1. Now we move on to our second session, which is going to be the panel discussion. And now I would invite Dr. Y.K. Gupta to moderate this second session. So Dr. Y.K. Gupta is the president of All India Institute of Medical Sciences, AIMS, Bhopal and Jammu. He's also the chair technical committee, Good Lab Practices, and vice chair national standing committee on medicine and healthcare products. He was also the ex-director of IITR, and he's been on several national and international committees and panels. So it's a great pleasure to have him chairing the next session, which is also a very relevant session on how funding can accelerate the development of these systems. So I would request Dr. Y.K. Gupta to please take it forward.
Thank you very much. Am I audible now?
Yes, you are.
Thank you. And before I start this panel discussion and invite the speakers, I must congratulate the organizers for taking up this wonderful and very important topic, webinar on such an important topic, which is contemporary, which is futuristic, and which is futuristic, and also which is so science and funding intensive that we have to debate it. And tomorrow, if you just think about, I will say that future toxicology and prediction of the possible evaluation of drug effectiveness, or how the drugs can be withdrawn, and what is the success, will depend on the results from organ on chips. So important is the topic. And it was a really treat to listen to Dr. Abhijit and Lorna on such a wonderful topic. Now, this panel discussion, I really am very happy that we have a very rich panel, very experienced panelists. And I will just give a brief introduction of the first panelist, Rajeshwari Adiseshan. She is SPO on chemistry, manufacturing and control vaccine development in Bill Gates Melinda Foundation. And she is alumni of BITS Pilani, which is one of the most prestigious institutions in India. And she has a 25 years plus experience in the biotech industry. Rajeshwari, before joining this foundation, she worked in major biotech firms in India, and as such as the Serum Institute, where she worked as a vaccine lead in CMC compliance and regulatory product development for several years. And her work experience is predominantly in product lifecycle management, formulation, regulatory, and quality. So welcome, Dr. Rajeshwari here. And Vishal, I know Vishal for many years now. Now I know Vishal as founder and CEO of BioRx Venture Advisors Limited and chief ideator, which is, I think, only high IQ people can do it, at India Healthcare Agencies, which is abbreviated as IHA. And he has an overall 22 years plus strong experience in investment, mergers, acquisitions, and doing due diligence process focused on healthcare and allied sectors. I thank you very much for joining us, Vishal. We
Thank
have
you,
also,
Gupta.
thank you, Vishal. It is always pleasure to interact with you. We have with us my friend Dheeraj Kumar, and Dheeraj is a chief manager technical at the DBT and in BIREC, and he is a senior science and technology management professional, and he has a 15 years plus experience in leadership and healthcare sector. Dr. Dheeraj did his PhD in molecular parasitology from a very famous National Institute of Pathology, which is an institution of Indian Council of Medical Research. And before joining BIREC, he was in US FDA and rich experience, and he is recognized for demonstrating a natural aptitude of biotechnology research as well as scientific project management and his contribution. And currently he is chief manager in BIREC, Department of DBT, Government of India, and responsible for products development, capacity building, and particularly capacity building in such advanced areas in indigenous development in India. And he also leads vaccine and R&D portfolio. Thank you very much, Dheeraj, for joining. And another
Thank you, sir.
speaker is Dr. Glenn Stacey. She is the Director, International Stem Cell Banking Initiative and UK Stem Cell Bank, which he developed as a world-leading resource centers for human stem cell lines. In 2007, Dr. Glenn also established the International Stem Cell Banking Initiative, which has developed to coordinate international pluripotent stem cell banks. He has also chaired the UK National Clinical HESC Forum for Scientific Advisory Board and Scientific Advisory Board for the Public-Private Partnership of a non-profit company called as a Stem Cell for Safer Medicine. Last but not the least, and she may be the first, Dr. Sonja Bacon is the coordinator of a unit of non-clinical evaluator at Belgian Federal Agency for Medicine and Health Products, FAMHP, and this unit is responsible for evaluation of non-clinical data submitted for support of all phases of drug development, be it market authorization, clinical trial, and national scientific advices, European Union, in European Union and in other places. She is also the chair of 3Rs Working Party in European Medicine Agency, which is an agency of European Union and in charge of the evaluation and supervision of pharmaceutical products. So we have 5 very strong panelists, and these panelists will give a brief what they can talk about, and then I will just put some questions to them, and I will expect their very focused answer, and then we can have the questions from the forum. And I have been told that Dr. Sonja Bacon is unfortunately not available with us, but she has sent her recorded statement. So may I request Surat to, if you can play her recorded statement to begin with, and then we can have the panel discussion, the questions to other panelists. Over to Surat to play the record.
Sure. Just wanted to play the question once, Usman. I just wanted to repeat the question once. So can you hear?
In fact, I just thought of what should be our questions to Sonja, because
So I'm just so the question that we had asked Dr. Sonia was if she could discuss 1 or 2 successful case studies where funding had led to a tangible impact on the development of organ-on-chip models, and also her opinions on what should be the focus areas for future public and private funding. So that is what we had asked her, and unfortunately she had another meeting which she had to go urgently, but she has sent her recorded statement on this question, which would definitely give us insights.
Great.
Usman, if you could play the video.
personally, when I think about a successful case of funding having a tangible impact on organ-on-chip model development, the Under the EC Horizon 2020 funded ORCID project comes to mind. This project had as a main objective to create a roadmap for organ-on-chip technology and the framework to build a network of relevant stakeholders. This project ran from 2017 until 2019. During the multi-stakeholder workshops that were organized under this project, important topics were discussed that lay the foundations for further discussions today. One of these, for example, pertains to agreements on context of use wherein these models can be utilized. The ORCID project evolved to the European Organ-on-Chip Society that is active today and will hold its annual meeting in July in Italy. From the European regulatory point of view, we aim for better prediction of both efficacy, ADME, and safety, and this through the use of better predictive models, be it in silico, in vitro, ex vivo, non-mammalian in vivo and in vivo, either alone, but most probably in combination, and or integrated in weight of evidence approaches. As such, new testing approaches should really focus on prediction and on filling data gaps, whilst in the same time addressing the 3 Rs as described in the EU Directive 2010-63. Regulatory acceptance of novel testing approaches is subject to context of use qualification. The context of use pertains to the description of the circumstances under which the testing approach is applied in the assessment of human or veterinary medicinal products, including a description of its limitations. This is described in the EMA guideline on regulatory acceptance of 3R testing approaches. The 3R's working party of the EMA is currently in the process of revising this guideline, and it's drafting specific annexes describing context of use qualification criteria for MPS and organ-on-chip models to be used in the pharmaceutical area. The initial focus is on predicting DILI and cardiovascular safety pharmacology testing endpoints. In addition, the EMA 3R's working party is revising the existing comprehensive inventories on animal testing requirements and opportunities for 3R's implementation for both human and veterinary medicinal products. These reflection papers provide an overview of regulatory gaps, needs, and future perspectives for introduction of new testing approaches, and could be considered a source of inspiration for context of use identification by multiple stakeholders. Now, whilst high-level guidance is available and more specific guidance under discussion, only a limited number of applications for novel methods qualification have been submitted to the EMA. In order to increase stakeholder interaction, the EMA 3R's Innovation Task Force procedure was set up in 2023, and this provides a clear entry port to a free-of-charge forum for early dialogue with regulators, and this for all types of stakeholders, academic centers, pharmaceutical industry, method developers, and large-scale consortia. In addition, to allow for sharing and evaluation of data generated by a novel approach, such as the MPS or the organ-on-chip model, the EMA allows for the voluntary submission of data generated through NAMs, such as MPS, along with the data generated through existing conventional methods. These data will not be used to make product-specific based regulatory decisions during this period, but this approach will allow the gathering of sufficient information on a novel technology before considering its possible regulatory uptake. The setup of such a process of voluntary data submission, or the so-called safe harbor, can be discussed under the ITF or later under the qualification of novel methodologies workstream. So how does this link to the funding? To achieve the ambition of having better predictive models in line with the 3Rs, be it MPS or other models, there needs to be a clear understanding of the regulatory needs and a translation to tailored funding schemes that also allow for the generation of qualification data. Interaction can be at multiple levels, being at call formulation, at the level of project-specific advice, and later on at the level of the appraisal of regulatory readiness of project outcomes. These different types of interactions could possibly increase the production of MPS models or organ-on-chip models that are ready for regulatory acceptance to a larger extent. And with that, I'm at the end of my intervention. I thank you for your attention.
Thank you. Thank you very much. Though you are not present physically, but I think the talk of Dunya was so clear and so brilliant. And she talked that for better predictive value, ultimately we have to get them in the regulatory approval process. And all this, I think, is requires a lot of funding process. And that is why the first question would be to Rajeshwari. Rajeshwari, you are working with BMGF, which is we look for the funding from BMGF on all such exciting and futuristic provision. How do you think that BMGF being a larger philanthropic organization, what are the criteria you think before you decide on the funding? And when you look for emerging technology, BMGF is known to fund emerging technologies, and this is one of the futuristic emerging technology with a lot of uncertainties and a lot of questions of the outcome. But we know that this will ultimately has to be translated into regulatory, which will be a total game changer. And but what do you think? Because now we have organ and chip for skin, organ and chip of placenta, but the major organs are still remaining to get validated. And for example, organ and chips on liver, organ and chips for cardiac, organ and chips for retinal damage, and so on and so forth. And so a lot of funding is needed for that. So what is your take on that? How you decide on that? How Abhijit and colleague like that should approach you?
Yeah, thank you, Dr. Gupta. Yeah, it's a very important question, and I'm glad to be part of this panel. So I can sort of tell you that typically what the foundation looks at for when it decides to fund an emerging technology. So I'm not going to be very specific on this topic, but generally when we are looking at an emerging technology, then there are some key criteria that the grantee can sort of expect when they approach us. One is that the technology must align with the foundation's core mission and goals. So what are these? The foundation, the BMGF, focuses heavily on improving health in the low and middle income countries. We have a focus on vaccines, drugs to a lesser extent, and the overarching goal is to provide equitable access, expand opportunities, provide more access to, say, information technology. Finally, the goal is that it should all come down to meeting foundation's principal goal of charitability. It is also crucial that the innovation, the product that comes out of the innovation meets our cost of goods requirement. So, the product should be similar, or better still, be preferably lower to the current, standards that are being used for the innovation. Other than that, the technology should have the potential to make a significant impact on the issues that it targets. This also includes scalability, because we are trying to reach a mass, population, especially in LMICs. So there should be scalability and also sustainability. Plus, there should be feasibility. So the foundation encourages manufacturers from the low and the middle income countries. So the manufacturer from, say, from India the Southeast Asian region, or or maybe Africa, should be able to have this feasible as far as implementation is concerned. Then, of course, it's easier to fund when there is already some proof of concept or evidence, or there is some sort of a package that has come out of, say, your pilot projects or some research studies or some form of validation. But having said that, the foundation also sort of gives funding to very upstream research as well. That could actually generate or work towards generating the proof of research, proof of concept evidence. And we also look at the team, the team that's working on the ability of it to sort of execute the plan, and if they have the necessary experience or the expertise and the resources. Because we don't want to fund something which which may not be sustainable, or which the company does not have adequate expertise on. And having said that, I think I mentioned all that. The approach should be that it should be sustainable over a long term and capable of scaling up. So these are the basic criteria when we look at any new sort of innovation. Specifically on this, this is a very interesting, from our speakers. You all may be aware that BMGF has already been funding towards the 3R, the acceptance. We want to reduce the animal testing as much as possible. So this could possibly be an extension on that current grants that we already have in place. And also there is a clear interest in foundation in having, demonstrating preclinical animal safety and toxicity, especially for vaccines, or maybe even adult vaccine use in pregnancy. So if there are, if these can sort of address these, then I think there would be, interest definitely from the foundation. Thank you so much.
Thank you very much, Rajeshwari, for giving the perspective of BMGF. You mentioned that you will be more interested, as per the guidelines of BMGF, in funding which are useful for LMICs and which are for the public interest and which have some proof of concept already. But you also are open for funding for the high science also. And I would say that this area meets both the requirement, because on one hand, you are funding 3R reduction or 3R philosophy, and this is one of the important aspect of 3R. Also, if you just look for the vaccine, but also not only vaccine, but also the anti-cancer drugs, the prediction of safety and the efficacy of anti-cancer drugs is a personalized medicine. This is one of the very important area, and I'm sure that this will be into your prominent area in your canvas for possible funding. So people should be very enthusiastic by your participation here. So thank you very much. I would now request Glenn to just, because Glenn, you have been establishing the stem cell bank. So how important do you think is the funding towards establishing the stem cell bank for the development of these technologies of organ and chips? Because these banks are again in the perspective of the common man. It requires convincing them, and it requires some sort of a risk, but ultimately this is the science of future. So what is your experience and expectation, Glenn?
Yeah, sure. Thank you for the opportunity here, Dr. Gupta. I mean, I think, having been involved in the stem cell banking activities for quite some years now, the banks are always interested to ensure that they provide good quality material that's suitable for research applications. And that's the quality control may be more general in terms of mycoplasma testing, identity of cell lines, and all of that is really crucial to the quality of the research that the researchers can use. If we just provided cell lines and didn't have any controls, people would be working with infected material, not using the correct cell lines, which is a common problem these days, and it's been reported on a number of times in the literature. An estimated hundreds of millions of research dollars and euros have been wasted on cell lines which are not what they were supposed to be. And this is particularly important for the IPS area, where we're working with banking disease cell lines for disease modeling, where we need to know that the cell line, we should know that the cell line has the correct disease lesion. So that providing those quality materials is an important driver. I think it's like an insurance policy for the researchers. They know that they're getting material that's been checked to a certain level, and they can check with the banks to identify what quality control they've done. That's very difficult to do with another research group, and it has been a common problem that sometimes the research group just in by mistake provide the wrong cell line to their collaborators, and it's very embarrassing. It wastes a lot of money and time, and it causes a lot of friction. And also, if you provide another lab with a mycoplasma contaminated cell line, and that's known to affect the performance of stem cell cultures, that also has a big effect on the lab. This mycoplasma problem can close down a lab for several months before they can properly clean the issue up and have acceptable material for publication. So I think that it's important to fund these banks and to emphasize to the population at large that the funding for these central centres, whilst it might seem quite large to put into just a place to hold the cells, they're not just holding the cells. They're making sure that they're available for the future. They're making sure they're of the right quality. They're making sure that the researchers can have a good starting point with documentation on the quality control that's already been performed. I think there may be some other things the banks could do, and maybe there could be some funding to help with this. And that will be, and this is something we discussed back in 2010 at the one of the ICBI meetings. And this was looking at the needs of the toxicology and disease modeling groups, and what needs, the additional needs that they have for the quality control of the cell lines. And it's something that was addressed within the European Biobank for IPS cells, EBIS, and they carry out tests for their disease-affected cell lines to check they have correct lesions, etc. But it will be also useful for the banks to talk together and to be able to do the work to identify appropriate panels of cell lines that will give good representation of different populations. And this is important particularly in the toxicology area, where regulators like the FDA for many years have said, if you're doing a toxicology test or a drug discovery test, you should really have representation of maybe 10 different genotypes. But then selecting what those genotypes are and how representative they are of different populations with an international recipient community of patients is very important to understand how representative those research materials are and test materials for industry. So there's a particular question on diversity of the holdings of IPS banks and ESL banks. There's a challenge for the banks to keep themselves funded and to just, I think the important thing for them is that they sustain those materials for long periods. They provide an insurance policy for the researchers, so they're much less likely to waste money on inappropriate cell lines or cell lines which don't perform, and to also for researchers to hold the cell lines they've generated in a safe depository, so that the researcher is not going to lose the material if there is a problem with their funding or with an accident with their systems. And these have been reported numerous times in the past. So I think, yes, there's a clear role for banks. That's an important role that the community needs to understand and recognise the importance of having this quality material maintained and sustained over the long periods of time for the research to develop.
So Glenn, I think what you have made it very clear that the whole world, and particularly in India also, we need to have larger number of stem cell banks. and that is the future. But more important is that people's faith on these banks, which largely depend on, as you have rightly said, the quality of the cell lines which you get and how you keep with them for a long time, and how the predictive value is that, and how variability can be established. And therefore, as you have very specifically mentioned, the millions of dollars of funding can go waste. And I would say not only the funding goes waste, infinite number of money, if you just monetize, of the faith of the public on that will go waste.
Yeah.
That is something which is very important, which you have highlighted. And that's why I will ask you, maybe in the next discussion, maybe now, that how a bank, how a funder will know that he is putting money into the right person or right organization who will take care of the quality and all those checks and balances. Because at the time of funding, what steps he will take so that these things are taken care of, checks and balances. So Glenn, maybe in a very short answer you can now, otherwise you can answer in the second round.
So yes, I can give you a quick answer, and I think there are communities like the ICBI who discuss quality and delivery of cell lines of pluripotent stem cell lines to researchers, and what the minimum standards should be for those biobanks. And we publish the reports from these conversations and sort of draft guidance for biobanks over since 2010, and we regularly have published discussions on these topics. So there is sort of an internal mechanism in the biobanking field which establishes those minimum standards, and you can ask the biobanks, do you address these minimum standards? And also the ISSCR has been working on some best practice guidance as well, and for more general pluripotent stem cell research, and that's also something you can refer to.
Great. Vishal, you are a wonderful ideator, and a lot of venture capitalists and the funders and the angel investors, they bank on your advice because you are so critical in giving them the advice. And so, and as Glenn has also said, that this is a bit risky if the quality is not, even there is an expertise, but they are even at 0.01%, they becomes casual at some place, or the naturally mistake can happen, and the confidence of the not only the the funder will shake, the entire thing may collapse. High risk but very fertile area. So what are your perception that these are the risky, and how you can increase the confidence of those who, private funders particularly? The government can usually fund it, but getting funding from private sector is tricky in this. So what is your advice, and what are you think that the funding seekers should look for? Over to you, Vishal.
Thank you, Professor Gupta, and thank you, Surat, for the opportunity. I've in my first leg of my career worked with Dabur Research Foundation and Ranbaxy, where I was leading the preclinical toxicology and other preclinical efforts. And I've now been looking at investments for about now close to 17 plus years. So I can truly relate to, the challenges that some of these startups or maybe some of the companies which are working on disruptive models, they're not able to mobilize money. And I think there are two intrinsic and two extrinsic factors that I can think of, which is across the life sciences area, and which could include drugs to devices to anything. So my comments are not specifically only to the preclinical, but what could we do to attract better capital, which is risk capital, to the biotech sector. So I think from that perspective, I would say that the entrepreneurs, are picking up such areas, and it's an interesting area. We've seen a lot of de-risking which BIRAC has done. In fact, some of the very interesting startups which have been funded by BIRAC is towards supporting disruption. But what happens when, those disruptions are not really moving to the next level of traction, from an adoption perspective, their ability to sort of get a regulatory approval, I think that's where, we get shaken. And so I'm going to talk about what shakes us and what makes us to say yes and what makes us to say no. So I think yes is very clear, because as venture capitalists, or as angel investors, we do know it's a game of high risk, high reward. And that is a sweet spot. So we got to fund companies which have actually high risk, because we want to get high reward. So there is no other solution than to fund high risk ventures. But when we are looking at funding high risk venture, that's also a time we need to sort of, de-risk ourselves. And within that de-risk framework is that to understand the domain expertise well. And today, all of us have expertise. All of us have external expertise to seek the sort of a value on the technology from the finance point of view. But we also see sometimes, that the regulatory infrastructure is not that great. And tomorrow, even if you have funded a startup, and tomorrow that regulatory infrastructure does not allow you to get into mainstream of drug discovery and development, and then tomorrow, the drug regulator says that this data is not acceptable, then, there is a problem. And this is happening in all the technologies, if you look at. So what we get to see that, one agency is funding it, and VCs are funding in, and angels are funding in, but the regulatory infrastructure in the country, which I can speak about basis my interactions with almost like more than 1,000 startups in the country across biotech area, for ranging from stem cell, I think Den was talking about stem cell, to molecular diagnostics, to genomics, to, think about anything. I think we do look at it, even agri-biotech for that matter, right? Agri-biotech is a technology, so wonderful technology, but we couldn't see the light of the day because there was no clarity on how the GMOs are seen from a risk perspective, and there was nothing one could do about it, although we have all the committees, but then still, we couldn't really harness the potential of BT technology or, agri-biotechnology. In the same way, I think if organ chips, I think it's a solution. I think there are many more models like organoids, and there are companies, even in India, startups which have got funded. But I think their further ability to raise capital will depend upon how the regulatory infrastructure around the drug discovery, and development is coming about. And sometimes we have seen that, we do try to emulate the model what US FDA has. So I think if we have US FDA model getting adopted in India, we do have a clear hedge mechanism to sort of, fund such startups, because we know that there is a need, because there's a clear need. The traditional drug discovery approach, I think, has not really worked. You just cannot have one molecule getting to the market after like 10,000 leads. We need to reverse that. We need to see that there is a signal, more signal than a noise. And how do we do that? So we got to anyway introduce new technologies, whether it's AI. We are in fact evaluating a very interesting startup which is actually trying to improve the outcomes from signal to noise, or NCE to the market, by leveraging AI. Now, how do we use such technologies in combination to the new technologies which are coming up, right? Organ on chip, I think, is a reality, is the future. But then, how does regulatory infrastructure sees it will actually depend upon, whether how much funding will come in. And I think I'm again giving a focus on regulation, because being a biotechnologist myself, and I've also been, like looking at startups, and being a, PhD in technology management, I understand that what it takes to develop a molecule to the market, and there is so much de-risking that has to be done from, preclinical stage A to preclinical stage B, and then later on human clinical trial 1, 2, 3, and then so much of resistance is there. Even big companies in India, I won't name it, but they find it also difficult to sort of take their innovation to the market, and they'll end up getting into partnerships, what I call as either M&A or in licensing, out licensing. There's enough structures, which with big companies are looking at, and that is why, one must look at that, why people are looking at partnerships, because one of the ways to how to address this regulatory challenge, I think a regulatory pathway is very clear. And this is where, Dr. Gupta, you being a scientist, a visionary, people like you managing such committees, spearheading these committees, I think needs to really, appreciate that the capital which is coming as a risk capital needs to see at least, clarity that, how, this would turn to be service, how this would turn to be a product. And that is where, the huge amount of money will come in, because this is the future. We just cannot avoid this. I'm sure the technology is the future, and whether it's a organoid or maybe organ-on-a-chip or some other new technology which actually helps you to increase the signal to noise, which means the lead molecule getting to the next stage. I think we just cannot think without this. It is the future, but we need people like you as key opinion leaders to help us de-risk that. And this is my very humble view. I'm not a toxicologist, but whatever little I know. But definitely on the financing side, I've been looking at various aspects, from debt to equity to hybrid to, even, we had Rajeshwari talking about, from a foundation perspective, which is very, very interesting to see that how Bill and Melinda, Gates Foundation coming and supporting such innovative technologies, because by then, BIREC would have picked up something, and we see that's another way to de-risk our own, funding. And disruption is the requirement. And again, within that, how do we de-risk from a private capital? Because in India, we haven't seen too many angels getting exits, the way one would imagine and one would like to. So how do we de-risk? That's what I probably would like to talk right now. And I wish, that we can really learn out of today's conversation and make sure that, we adopt known animal models. And I think that is the future. We just cannot think anything without, non-animal. I think this is where I would like to end for now and pass it back to Dr. Gupta.
Vishal, I think you have touched my heart, because what I would have said, I think you have said it. And so,
Telepathy, sir.
So correctly and so aptly, in the drug development, we used to make a slide which we must replace. Slide means when you teach the drug development process to our M-Pharma, PhD or pharmacology students, that you have to have 10,000, maybe or more, and then you may get one drug to develop. I think that is what the noise to signal ratio has to change. And organ on chips is the answer, where you can just reverse this noise to chip and noise to signal. There will be signal, more signal, and reduction in the noise. The
Absolutely.
efforts, all money will go waste unless this validation does not take place. If this validation takes place, and the valley of death of all those would be if they are not approved, accepted, or adopted by regulators. That is also, you have very rightly expressed. Regulators will do it only when it is written black and white in their regulation. Incidentally, or unfortunately, the regulatory science in India is almost non-existence. So we have to have a training of regulators where they can also think out of box, not always copying from OECD or anywhere else. See, and therefore continuous interaction with the scientists, funders, toxicologists, drug development, pharmaceutical industry. And that's why I always believe, and I have been propagating, that early involvement of regulator in such is important. With this background, we are also having in plan, as a president of Alternate to Animal Society, a platform where the regulator and these researchers can be interacting right from the beginning. So maybe once it is freezed, and we will invite you all. So, but very important, which you have said, that it is the risk, high gain, but this risk has to be some de-risking. Who will do de-risking better than the BIREC? See, BIREC is the organization which is promoting Indian pharmaceutical development, disruptive, as you said, disruptive innovation, futuristic innovation, and funding is also to startup promotion, and also what you have rightly said, to partly de-risk. But the success of BIREC is always estimated when these startups become independent unicorn. And that's what the BIREC, and I am very happy to see Dheeraj here. And Dheeraj, just tell me, BIRAC has released recently a call for proposal and such technologies. And what you think, in your view, the current landscape in India for such things? And is there a scope for separate funding program for these methods in India? Recently, I was talking to one of the investigator in JNU, Dr. Rana, and he may be, I don't know whether he is listening or not, and they are looking for BIRAC first, that BIRAC हमारे पास सबसे पहले आना चाहिए. Let us first explore BIRAC because they are very futuristic, progressive funding. So what is your take, Dr. Dheeraj?
Thank you, sir. Thanks, Dr. Surat, to organize this wonderful talking talk. So, sir, BIRAC is supporting this kind of studies from very beginning, and from through our different scheme like BIP, CDRI, PACE, and all. But we feel that still that only 7 proposals in this area came to us, and we supported. So, like in this 7 area, the leading proposal was from ICT Mumbai. It is on lung-on-chip. Then another was on tricomponent lung organoid model. Then a dengue virus mouse model, and that NAS model, and 2D interface model of lung airway and alveolar epithelial cells model. Transgenic mice SARS-CoV-2 COVID model for in COVID therapeutics, and dogs inducible S2 transgenic model. So only this 7 proposal we have received through our regular call. So we felt that there is a need of this kind of model or this preclinical model. So that's why we announced a special focused call on the development of preclinical disease models for screening new drugs, NC, natural compounds, vaccine, etc. And with this initiative, now we are in the process of supporting 10 technologies or model in this area. But this will not end here. Means we are not going to end at that, okay, now finish, we have announced a call. We are still feeling that means many of the researchers not came to us, and maybe they're not aware with that BIRAC is very keen to promote this kind of work. So we are going to announce another round of call in this area. So we are hoping that in that time we will get more proposals. So means there is fund, and BIRAC is always ready to support in this area.
Thank you, Dheeraj. I wish there are more Dheeraj in BIRAC, because the positive approach which you have shown to the researchers in this area is really very heartening. And the disheartening thing is that I asked Surat just a day back that do you have the database that how many centers in India are working on organized? Because I was aware of only few, and she said more than 70. If they are more than 70 and only 7 proposals are coming, it indicates that they need to be motivated.
No, there are 70 and more also. But before announcing this special focus on preclinical model, BIRAC was supporting only 7 proposals. So that's why we felt that there is a need, and people are also working. Then why maybe some restriction? So we announced a special focused call with a special advisory committee from this specific area. So now we are in the process of supporting 10 more technology, but still we want more. That's why
No,
we are
I
going
fully
to announce
agree.
another round.
I fully agree, Dheeraj, with you. But my caution, as previous speaker has also said, that support only those where you have a faith and trust and monitoring, because as this is a very emerging area, and if just people say that this is the low hanging funding possibility and they don't take it seriously, I think we will be a loser. So I really appreciate the BIRAC's effort in this, that this is an emerging technology, 3R factor, and more importantly, developing a drug at a lesser time, lesser cost, will have a very high direct impact, and personalized medicine, particularly for cancer and vaccine. I think this is a inevitable, and the future toxicology will be based on organ on chips or organoids. And but as Vishal has said, that BIRAC, I think BIRAC would be the appropriate agency which can invite the regulator and such forum. Maybe you can sponsor few workshops. Maybe I would be approaching you to sponsor workshops on this, where regulators must participate, and I will ensure that the scientists may participate. And the idea should be how to ensure the translation into the regulatory acceptability of such wonderful ideas. So thank you, Dheeraj. And I am really happy and very thankful to all the panelists for giving. And this discussion could have gone for 2 more hours easily, but because of the shortage of time, Surat, will you permit
Yeah.
some questions? My computer is not able to see it, but if you can read from on my behalf, it will be very happy.
Yeah, I mean, there are one or two questions in the chat box, but we are really overshooting the time. So I would request the panelists, if they can respond to the questions in the chat box to save the time. And I would now request, thank Dr. Gupta for, conducting this session so beautifully and also giving your insights during the process. I mean, they were very helpful for both conducting the session and for all the members who are on this webinar. So again, I would like to thank all the panelists and Dr. Gupta. And now maybe we can just end the session. And I would like to request Alok Parna Sen Gupta, who is the MD of Humane Society International India. And I would invite her to give the closing remarks. And yeah, thanks. Over to you, Alok.
Just before Alok Panna, you say, my last advice would be to you, Alok Panna, and all the organizers. This should be the beginning of your effort. Continue this effort, organize such webinars more frequently, invite regulators more intensively, and I think we will be successful in our getting whatever we want to have. Thank you very much. Over
Thank
to
you
Alok.
so much, sir. All right, I'm going to quickly share one slide for just talking about where we want to go from here. We would want to continue, like Dr. Gupta said, we would want to continue this engagement and momentum which has been created to further the discussion. So towards this, I would like to invite you to join us as we plan several initiatives, a few of which are facilitate the development of guidance documents, resources, and publications towards NAMs, non-animal methodologies, establish multi-stakeholder working committees on priority disease areas which for India, and which will help Indian science, Indian public health, and of course develop national and international funding proposals to increase investment in NAMs, not only in India but also globally. But of course, our priority is in India. So please, those who are listening, those who are part of the panel, those who have spoken with us on this, please reach out to Dr. Harshita Mittal and Dr. Surat to learn more about this and to collaborate with us. Now quickly, because I think it has been such a good conversation, and I know that we could go on for a lot more time, but it is my honor to be able to give a vote of thanks, firstly to Dr. Y.K. Gupta. It's a moment of pride for me that he comes to a webinar which is hosted by an organization I work with, because when I started my career in in an NGO for replacing animals in animals in experimentation, he was one of the first few people I met about 15 years back. So it's really a moment of pride for me to have you on this panel, sir. Your leadership has been and will continue to be very essential, and we look forward to your advice in the future as well, including making the connection between academia, research, and industry and regulators. Of course, our esteemed speakers in the beginning, Dr. Lorna and Dr. Abhijit Majumder. Again, Dr. Abhijit Majumder is somebody who I've met many years ago first at IIT Bombay for so simply, both of you so simply putting such complex topics together. Like that's where we understand, right, how good scientists you guys are, because you're talking about liver on a chip, placenta on a chip, and it's so easily understandable. So thank you for that. Again, fascinating to learn the potential of these. Thank you to our panel members, Dr. Rajeshwari from Bill and Melinda Gates Foundation, Dr. Dheeraj from BIRAC, Dr. Glynn from Stem Cell Banking Initiative, Dr. Vishal Gandhi from BioRex, and of course, Dr. Sonia, who wasn't able to be here, but she did present her thoughts. You guys presented very, very unique and pertinent points as to how we could accelerate NAMS. And I, again, taking from Dr. Gupta's this is only the first of the many conversations that we want to have. Our partners, BIRAC, STEMPeers, and CPHMS, who stood strong, and without them, we won't be able to do this. To our audience who came in, now they are dwindling because we are over time, but we had over 70 participants here, and who stayed on for almost 2 hours. So without them, there is no point of doing a webinar. But I think none of this would be possible without Dr. Harshita and Dr. Surat, who are part of our research and toxicology team. So kudos to you. To Moshumi, who leads the government affairs program here. Of course, Helder, Troy, Kate, Francesca for our global team for their vision and leadership. And of course, our team in the background, who helps us be tech savvy. So Usman for being there throughout to help us do everything. Navmita, Kabani, Sushmita and Shelly, Aviva, Marta, Uma, and Uma, who are a part of our research and toxicology global team, who help us with the science, with the communication, and so much more. So I would extend an invitation to all of you to join our initiatives and help this initiative reach its ultimate goal of not only animals being replaced or reduced, right, for better science, better public health access, and a better country in the future. So thank you so much for your patience and for your participation.
Thank you.
Thank you.
All
And
right,
thanks
thank
to
you so
Alok.
much.
Thanks to Alok Parna for overall guidance and support to make this happen successful. Thank you to you.
Thank you so much.
Thank you, everyone.
Bye-bye.
Thank you, everyone. Bye-bye.
Bye.
Thank you. Bye.
Thank you.
Bye-bye, everyone. Thank you.